Hi, everyone. So just again, if you could just keep yourselves muted as we go through this session, we would greatly appreciate it. My name is Dr. Haggerty. I'm with Thomas Jefferson University and one of the co-founders of Anato-Bee. You usually see Dr. Pasquira introducing everyone. Tonight, I will be coming in and introducing this session. So this is Anato-Bee's 7th tutoring session. We're going to be looking at some GI and anatomy, histology, and embryology. And before we get started, we like to do a couple of announcements. So again, I see we're still admitting people. Please keep your camera and microphone off if you choose to not be recorded, because we will post this on our YouTube channel. And we want to welcome you all back from Thanksgiving break. So we hope you all had a wonderful holiday break. You got some time to relax, and we're very thankful to have you all here for us with us on this Sunday. So again, this is session number 7. There are 3 more sessions after this. So you'll have 2 more sessions with our lovely medical students tutoring you, and then you'll actually have a review session with some of the faculty to go through an overall review before the competition. Our next session is going to be 2 weeks from now. That's going to be session number 8 on reproductive and urinary system, and that'll be December 15th. And then just remember, we're gearing up for the local competition. So the competition itself is going to be on January 25th. That's Saturday, January 25th. And it is going to be based off of the local objectives. So whenever you go in and look at our objectives online, and maybe I did have this up for everyone. So if we go into these, can you guys see my screen? Okay, so if you go online, here's what the tutoring schedule looks like, but you can go to our resources, you can go to the learning objectives, and when you click on any of these objectives,
Dr. Haggerty, I think I'm seeing the wrong screen. I think I'm seeing just like full
yeah,
files that you have.
all my files. Oh no. Okay, well,
There
I,
we go.
I picked the wrong screen, everybody. I hope you got a good look. So let's start again. If you go to our website, go to resources and our objectives, and I know all of you are probably pretty familiar with this, but just as a friendly reminder, you click on one of the objectives, it'll pull them up. This is for histology, but remember we have anatomy, histology, and embryology here. You can see on the left column here, those are the local objectives. So these are the ones we will be testing you on in the local competition on January 25th, and that's Saturday. So basically the only other announcement I have is that if you have any friends that are interested in registering, let them know last day to register is going to be January 13th at 12 a.m. Eastern time. So that's January 13th, 12 a.m. Eastern time. Before we get started, any other announcements? All right, amazing. Dr. Frank, if you could kick it off for us.
Hey, welcome back, everyone. Our first presenter this evening is Calista Mueller. She's going to be presenting on the anatomy of the digestive system. Calista is from Nashville, Tennessee, and graduated from George Washington University in 2023 with a major in public health and a minor in biology. She is currently a second year at the Edward Via College of Osteopathic Medicine in Auburn. In her free time, she enjoys going for walks, spending time with friends, and reading good books. So far, Callista's favorite part of anatomy lab has been learning neuro and cardiac anatomy with her awesome nerve anatomy lab table mates.
Okay, I'm going to share my screen and get the presentation going. Okay, can everyone see that? Okay, like Dr. Frank said, my name's Calista, and I'm really excited to talk to you guys today about GI and digestive anatomy. So these are some of the learning objectives that we're going to cover today. We're going to talk just a little bit about the larynx and pharynx. We're going to focus most of our time talking about the foregut, the midgut, the hindgut, and we'll also talk about the major organs there and also their blood supplies. And then we're going to talk about some of the accessory organs of the digestive system, and then also the liver. Let's see. Okay, so to kick it off, we're just going to talk basically about like what the overview of the digestive system does. So I kind of want you to think, just like a thought exercise, think back to the last thing you ate today and think about how those large pieces of food were broken down into nutrients that, your body was able to process and use to function and move your body along. So the digestive system is really amazing, and it helps nourish our body with a constant supply of nutrients, water, electrolytes. It's really important for the motion and movement of food through our body, as well as things like absorption, storage, and even helping pass waste. I think the best way to break down the digestive system is looking at both the alimentary canal and then the accessory organs. So your alimentary canal is where the actual tubal structures that the food is going to be passing through. So we have things like your esophagus and your stomach and the intestines, whereas your accessory organs are going to be organs that are helping with digestive processes by releasing things like enzymes and juices to help down your food products, but those don't directly have food pass through them. The major motion that we see for moving all this food along in the digestive system is something called peristalsis, which is a slow wave contraction that's going to propel a food bolus. I like to think about it, if you have a bit of toothpaste at the end of your toothpaste tube and you're squeezing it, you're pushing out the muscles are contracting at one point to move it a little bit along, and then those muscles will relax and the next set of muscles kind of contract to move it along. So it's like a wave of contraction that helps move the food down through your alimentary canal. And this is an involuntary motion. And then often your digestive system, we talk about it like clinically in terms of the 4-quadrant system. So up near your liver, you have your right upper quadrant. Over here by your spleen, you have your left upper quadrant, and then you have your right lower quadrant and your left lower quadrant as well. Okay, so this is kind of just a little table that we put together to talk about foregut, midgut, and hindgut. And in embryology, I think they're going to talk a little bit more about where these terms kind of come from and how we get them. But basically, what you need to know for the anatomy of it is foregut is the beginning part, and then midgut is the middle, hindgut is the end. So we're going to focus on the foregut first, which goes from your esophagus to the first part of your small intestine and the duodenum. And then we'll talk a little bit more about what it does, but main function here is we're thinking protein, carbs, and lipid breakdown. And then also your foregut includes some of your accessory organs like your gallbladder and liver that help with bile production. Okay, so just like a quick overview of the larynx and pharynx. Again, someone will talk about this a little bit more in depth later, but this is where your digestive and your respiratory systems intermingle. So your pharynx has 3 portions. There's this part up kind of in line with your nasal cavity that's called the nasopharynx, a part right below it called the oropharynx, and then your laryngopharynx. And the larynx is really important for connecting to your trachea, and then the pharynx to help with airflow and to make sure, so we want to make sure when we're swallowing that food doesn't go down the wrong pipe. So we have this handy little flap of cartilage. It's called the epiglottis, and it just covers up the trachea, and it actually prevents any food from going down that way and directs the food instead down the esophagus. Okay, so speaking of the esophagus, we're going to start there. This is the first part contributing to your foregut. It starts at the distal end of your pharynx, and it actually has some important anatomical things that we talk about quite often in lab and stuff. So relationship-wise, it is posterior to both the larynx, the trachea, and also the heart. So if you look at this image down here of the cadaver, basically where you see this thoracic aorta, if you follow that up and you just imagine the heart being right there, it's actually in front of the esophagus. And the esophagus itself is basically just a large circular muscle that uses peristalsis to move your food bolus down into your stomach. It has 2 sphincters. There's both an upper esophageal sphincter right around here, and then a lower esophageal sphincter closer to your stomach. The upper esophageal, and both of them just help prevent backflow of food. And when those don't work, you can get something like GERD or gastric reflux, where you feel kind of that acidic burning in your mouth because the food is kind of came back up. And then some kind of a important anatomical relationship is we often talk about which level different structures enter the abdominal cavity at. So the esophagus enters the abdominal cavity at the level of T10. And a fun little mnemonic to remember that is the I ate 10 eggs at noon. So I8 stands for the inferior vena cava being at T8. The 10 eggs is where we're talking about the T10 level for the esophagus. And then at noon is for the aorta at T12. Like I said, we're focusing on the esophagus, so that's going to be the 10 eggs part. So yeah, it's kind of a fun little mnemonic. And then the last part of the esophagus is the gastroesophageal junction area. And this is where you have that lower esophageal sphincter, and that's what controls the food bolus entry into your stomach. Okay, so now we're in your stomach. Your stomach is part of your foregut, and it has 4 main regions to it. The first, right up near this lower esophageal sphincter area, is the cardia of the cardia or the cardiac of the stomach. Next, you have your fundus of the stomach. Then you have your body of the stomach, and then your pylorus of the stomach, which is the lower part down here by the pyloric sphincter. The stomach also has 2 curvatures to it that are important because they have blood supplies that run along them. So you have the lesser curvature and the greater curvature. And the main function of your stomach is to store your food. It's for motility and digestion. So even like when food drops into your stomach, there's a motion going on it pretty much all the time to help mix up that food. So you're getting both a mechanical digestion with the mixing waves to create the chyme, while you're also getting some chemical digestion by release of different gastric enzymes. There's bunches of specialized cells in your stomach that help with that. And then also an important structural feature of your stomach to help with digestion is it has these submucosal folds that are called rugae. And these increase the surface area of your stomach and also help with the motion of digestion there. And then at the bottom of our stomach, as we transition into the first part of the small intestine, the duodenum, we have another sphincter. This one is called the pyloric sphincter, and it just controls stomach emptying to make sure we don't have too much movement or too much at a time release into the stomach or into the small intestine. Okay. And then kind of a little sidetrack from our alimentary canal that we've been talking about, we're now going to talk about an important accessory organ for the digestive tract, which is the liver. So the liver's main job is it helps produce bile, and bile helps emulsify fats, which emulsifying the fats just means you're breaking down the fats into smaller globules that can more easily be digested by our body, because fats are kind of hard to digest on their own. So the liver is really important. It has a ton of blood supply that goes through it. And those vessels are huge when you see them like on a cadaver, and you as well. But that's kind of like from this picture down here, you can see how big all 3 of those vessels are. And it acts as the body's filtration system. So it's really important for removing toxins and substances. And it also helps absorb nutrients back into your bloodstream, which is really important too. Some bit about the anatomical relationship of the liver. It's going to be right inferior to your diaphragm. So if you were to palpate the liver, you kind of go to the right upper quadrant and you can have the person breathe in and out and you can kind of feel the liver right there. And it's on the right side of your abdominal cavity. So it's in your right upper quadrant. And there's kind of 2 ways of breaking down the liver to look at it and study. There's the 4 anatomical lobes that we would like identify in lab. These are things like the right lobe, the left lobe, this small caudate lobe in the front, and the quadrate lobe. And then also there's a classification system called the CONOD, I believe I'm saying that right, classification system. And that breaks the liver into functional lobes, which are important for, clinicians, especially like surgeons, because each lobe of the liver actually has its own inflow, outflow of vascular supply and biliary supply. So this is how they can remove a part of a person's liver and not have to take the entire liver out, and you still have functioning liver left because they each have their own functional lobe. So moving on, we're going to talk about the gallbladder now, which
before you move on, can we go back to that previous slide?
Yes, ma'am.
Can you just clarify what right upper quadrant means? 2 things, clarify that, and then so that image on the left, I really like it because I agree it shows all those big vessels going in, but that's not how the liver sits in our body. So if you could explain what the right upper quadrant where that is. And then kind of explain a little bit how the liver is oriented more so in that image on our right here versus how we're seeing it on the left.
Yes. Okay. So right upper quadrant is kind of when we were talking about earlier. If you were to divide your stomach into 4 parts clinically, you have the right side, the left side, and then you can split them into quadrants too. So that's what the clinician is doing when they come to palpate your stomach to see where the pain is. They'll palpate first in that right upper area, and the main organ there is your liver. And then like Dr. Haggerty was pointing out, the liver isn't oriented in the way the image on the left shows. It's on the right. So if you see the image on the right, if we imagine across the top right here, you have the diaphragm and then you have the liver. So if the liver is my fist, the diaphragm sits right on top of it. And then those vessels kind of go through it like a straw, almost goes through a cup. And those then pierce through the diaphragm. Was that good for clarification? Okay. So now we'll talk about the gallbladder. And I think I mentioned this earlier, but just to better clarify what an accessory organ is it's an organ that's really important for digestion, and we couldn't do digestion without it, but it doesn't actually have the food pass through it and, help with like the actual mechanical digestion part with the food passing through it. So these just secrete digestive enzymes. And the main function of the gallbladder is it's what holds, concentrates, and releases your bile. So I know it always looks green in pictures and people are like, is it really green? It's really green because your bile is green. So it's kind of a fun fact. And it is going to be, if we imagine so this is our diaphragm, this is our liver, our gallbladder is going to sit just on the inferior surface of the liver down there. And it's kind of like tucked up in it, like a little, it's much tinier than the liver, but it's tucked up in the bottom of the liver. And it actually has 3 regions of it. There's this area in the middle called the body. You have the fundus at the top, and then the neck, which then goes and connects more towards the 2 ducts. So when you're eating something, especially like a fatty meal, your gallbladder, it's stimulated and hormones are secreted to send stored bile from the gallbladder to go into the duodenum to help with emulsification, which is just that breakdown of the fats. So they'll travel through, the gallbladder will get the stimulation to secrete bile. The bile will go through the common hepatic duct into the common bile duct, and then it will enter into the small intestine where it'll release bile to help with that emulsification. Okay. So now we've talked about the bile entering the small intestine. We'll get into the small intestine. So this is actually where we transition from being in the quote unquote foregut to the midgut. So the foregut is going to be at it ends at the first half of the duodenum, and then the midgut is kind of the rest of the small intestine there into part of the large intestine. And the main function with the small intestine is to just help finish up your digestion and absorption of nutrients, and especially water absorption happens here as well. There's 3 parts of the small intestine. You have your duodenum, which is the purple part up here, the jejunum, which is this greenish blue part, and then the ileum, which is this like pinkish red part. So the duodenum is an upper portion. It actually runs right next to the liver, if you can imagine the liver over here next to the stomach. And it wraps around the head of the pancreas, which is another accessory organ we'll talk about in a little bit. And there's the spot that we were just talking about where your bile enters into the duodenum to help with that digestion and the emulsification of fats. That specific point is called the ampulla of Vater. And that's where the pancreatic ducts and the common bile ducts come together. They empty that bile and other pancreatic juices into the duodenum. That way you can have the mixing and the digestion, emulsification of fats. And then your jejunum, it's a middle portion. It's mostly located in your left lower quadrant, or left upper quadrant, sorry. And it also just helps finish digestion and absorption. And then your ileum is actually the longest portion, and it is what terminates into your cecum, which becomes your large intestine. And then kind of just throughout all of your small intestine, you have circular folds, villi, and microvilli that help increase absorption and surface area. That way your small intestine is able to absorb all those nutrients and water. Okay. So speaking of the pancreas and the duodenum wrapping around the pancreas, we're just going to talk a little bit about the pancreas, which is also an accessory organ. And its main function is to produce these things called pancreatic juices, which are just fluid concentrated with digestive enzymes. There's a bunch of different kinds, and you can get more into the specific weeds of that. But the main portion you need to understand is that they empty into the duodenum with the bile from the gallbladder, and they help break down nutrients. And so the duodenum or the pancreas has it kind of fits right along the duodenum. If you imagine the top of the duodenum right there, it kind of wraps right around the head. And then the tail goes all the way out and kind of tapers right by the spleen. So it kind of stretches across both your right upper quadrant and your left upper quadrant. And the pancreas is a unique organ because it has both exocrine and endocrine function. So the exocrine function is the secreting of the digestive enzymes, and the endocrine is releasing hormones into the blood, which I think you guys will talk about endocrine stuff more in the future. And just down here, I kind of outlined the pancreas portions on this cadaver image, but your head is the part that's nestled into the duodenum. The tail is the part by the spleen over here. There's the uncinate process, which is also kind of closer to the duodenum stomach area, and then your body is what extends between. Okay, so I have some poll questions. The first one is, when you're talking about the GI tract, what part is the most important for nutrient breakdown and bile production? Okay, I think I'm seeing a lot of answers, and most of you guys are right. It is the foregut A. So that's really important because the foregut is right where we said that your gallbladder is a part of, that's producing your bile, as well as part of your small intestine, like the duodenum. That's all part of your foregut. So good job there. And then for the second question, we have a patient presenting with reflux symptoms after they eat a big meal. They kind of get heartburn and acidic taste in their mouth. What sphincter do you think could be causing that problem? Okay, I think most people have answered it now, but great job again, seeing most people picked B, the lower esophageal sphincter. And like we said, that makes a lot of sense since the lower esophageal sphincter is what opens up and is going to allow the stomach or the contents from your stomach to go into your esophagus. So that would definitely contribute to that kind of heartburny acid taste in the patient's mouth.
Melissa, before you move on, it's Dr. Peterson. Maybe just for those that don't understand what a sphincter is, could you just describe it to them so they have a better understanding?
Yes. Oops, sorry. A sphincter is basically just a closure around, it's a muscular kind of closure around part of the organ, so like in the esophagus, and it opens and closes at certain times to allow things to flow through it. I guess not things, the food bolus. And so your sphincter, and it's normally controlled by like kind of neural control or, pressure and stuff like that. And so the lower esophageal sphincter will open and allow stuff to go down through into the stomach. But if it stays open for too long and some of the stomach fluid pushes back up into the esophagus and then it closes, that fluid has nowhere else to go back down. So you'll get reflux. I don't know if anyone else has something to add there to make it a little more clear. Good. Okay. Okay, so now that we've kind of tackled the foregut, we're going to move on to the midgut and the hindgut. So The midgut is basically going to run from the distal part of your duodenum all the way until the first 2/3 of your transverse colon, whereas your hindgut is going to run from your transverse colon all the way down to your anus. They both have different blood supplies. We'll talk about those a little bit later. And the function is kind of very similar to the foregut because all these things are absorption, but we have less actual breakdown going on here and more just absorption. So in the midgut, we're going to get absorption of nutrients like your vitamins, your minerals. And then in the hindgut, it's going to be absorption of water and electrolytes. And then also that process of storing and compacting for waste. Okay. And so for the large intestine, I thought it would be easier to break it down via chart. So your large intestine is largely made up of the colon, the rectum, and the anus. Your colon runs from your cecum all the way to your sigmoid colon. And I have a picture of that later that'll show it a little bit more clear. And your colon is where all your gut flora and microbiomes are, which is pretty cool. It's important for waste storage, and it's kind of the last place that we get any sort of absorption. By the time you're moving into your rectum, we're more talking about storing the waste and the fecal matter, and you're not really absorbing anything anymore. And then your anus is mostly important for waste removal and your voluntary bowel emptying. And you also have some sphincters there as well, kind of like we talked about with your esophagus to help control your voluntary movement of a stool. Okay, so now we're going to talk about the specific regions of the large intestine. So I kind of outlined them over here in a color, so hopefully that kind of allows you guys to follow along with me as we talk about them. But like we said, the main function of your large intestine is to absorb water and electrolytes, and then also for storage of waste prior to going to the bathroom. So your 4 regions here are you have you have your cecum that you start out with here, then you move into your ascending colon. You have a place called the hepatic flexure, which hepatic is just another word for liver. So it's right by your liver, it's the corner. And then you have the transverse colon. So this is all midgut. And then about 2/3 into the transverse colon, we switch to the hindgut. Then we have this area up here, which is called your splenic flexure, which just means it's by your spleen. So you have that curvature. Then we move into your descending colon. You have your sigmoid colon, which is pretty uniquely looking. It kind of looks like an S if you look at it. And then you move into your rectum right there. So the large intestine doesn't have circular folds or villi like the small intestine, but it does have lots of mucus that helps with moving the waste products along. And instead it has these kind of unique anatomical things you can see on it called haustra and tenia coli. So the haustra are like tiny little outpatchings of the colon. So you can imagine kind of like small little, kind of like grape-like pouch hangings of it. That's kind of what I think of when I think of a haustra. And then the tenia coli are just bands of smooth muscle that go around the length of the colon. And those are both just important for helping with movement as well. And then now we're going to move on to talking about blood supply. So for blood supply to the foregut, midgut, and hindgut, they're all coming off of branches of the aorta. So I have this picture here. And for the foregut, I want you to think of the celiac trunk. That's kind of the main branch that we're going to talk about, and then some surrounding branches. For the midgut, I want you to think about the superior mesenteric artery. And then for the hindgut, we're going to think about the inferior mesenteric artery. So I'll get a little bit into that. And so for the foregut, like I said, our main blood supply is going to be the celiac trunk. And then off of the celiac trunk, as you can see over here, we have 3 branches. We have the splenic artery, which runs kind of right along the pancreas, the body of the pancreas, down into the spleen. And it also has 2 branches. And the splenic artery is going to supply your pancreas, which makes sense because it's running right along the body. Your stomach, it's got the posterior fundus and the left greater curvature. And then your, oops, sorry, I clicked that too early. Your left gastric artery, that's going to kind of run right along the area near the stomach there. And it's going to supply your esophagus and your stomach along the lesser curvature, which if you guys remember, that's kind of that the semicircle, I guess, kind of round moon shape along the top of your stomach, and the greater curvature is that one along the bottom of your stomach. And then your common hepatic artery is going to go to the right side over towards your liver and your gallbladder. And so it's going to help supply your liver, gallbladder, the right side of the lesser and greater curvature of the stomach, and then also the duodenum and the head of the pancreas are over that away. So these small individual branches just kind of are named after the things that they're supplying. So we have those branches are going to supply those areas. So for example, gastroduodenal is going to help supply the it's going to help supply the duodenum, and also the celiac trunk is the very first branch off of the abdominal aorta. Okay, so now moving into blood supply for the midgut. We're going to be thinking the superior mesenteric artery, which is number 8 over here on this image. And so it's going to have a bunch of branches. The main ones I think are important are you have your middle colic artery, which is going right here with number 6, and it's going right towards, if you look at it, the ascending colon, or the transverse and kind of this oh my God, hepatic flexure area. So that's what your middle colic artery is going to supply. And then if you look right here at number 4, that's going to be your right colic artery, and that's heading right towards your ascending colon. So it's going to supply your ascending colon. And then down here, number 10, we have your ileocolic artery. It's heading down towards that area between your ileum and your colon. So it's going to supply your ileum, your cecum, and also there's an appendicular artery that runs to your appendix. And then you also have this inferior pancreatico-duodenal artery, which goes and helps cover the pancreas and the duodenum, and it actually forms an anastomosis with one of those arteries from the foregut that we had talked about. And then you also have intestinal arteries. So these are just branches that help go to both your jejunum and your ileum and help give them blood supply. And then an important thing for the arteries when we're talking about the SMA and the inferior mesenteric artery is they help give this branch called the marginal artery of Drummond, and this creates an anastomosis to cover an area that doesn't get a lot of blood supply. So if you look up here in the left area of the screen, that's where your marginal artery is going to cover, because your IMA covers all these branches down here, but this area of your stomach, it almost kind of creates an area that doesn't have enough blood supply. So that's what that marginal artery of Drummond helps cover. Okay, and so now when we're getting into the hindgut, like we said, the blood supply here, we're thinking inferior mesenteric artery, which is this one labeled IMA over here on the image. And this does give branches that help form the marginal artery of Drummond, which kind of helps form that anastomosis to give that left upper area of your colon blood supply. And then it also has its own branches. So there's the left colic artery, which is this LCA here, that supplies the descending colon. Then you have your sigmoid artery, which is going right down here towards your sigmoid colon. So that's going to supply part of the descending and the sigmoid colon. And then you have this superior rectal artery, which is going right down here. And that is going to supply the proximal area of your rectum. And again, IMA is also a branch of the abdominal aorta. Okay, so I have a couple more questions to help nail in that blood supply, because I know it can be confusing, but.
Right, starting with the first one, Calista, can you read off that first question?
Oh, yes, sorry.
Yeah, no worries.
Brain fart there. So for this question, we're talking about what 2 arteries help supply the anastomosis that covers that marginal artery of Drummond.
I'm trying to remember too if we've covered anastomosis in our previous sessions, but if we haven't and you guys were confused about that, it just means that there's a communication between two arteries, creating some possibility for blood to come from multiple sources.
Yeah, sorry, I should have explained that a little bit better, but to me, anastomoses make the most sense when you think about there's an anastomosis right by your elbow and then also your knee. And it's really helpful for when like your arm is bending, if certain arteries may get compressed by that motion. And so others can help fill its place so you still get blood flow. Okay, so it looks like a couple of us got that, yes, it's going to be our superior mesenteric artery or our inferior mesenteric artery. There were some people that were thinking it was the celiac artery. But let's just back up and look at that picture maybe one more time. That first picture. Here. Sorry, I have so many transitions that's making it go a little slower. Okay, so okay, this one. So your celiac trunk, if we remember, that's the first branch coming off of the abdominal aorta, and then your superior mesenteric is going to come off down here and the inferior mesenteric down here. So celiac trunk, like we said, is kind of doing more of this kind of area by your stomach, your duodenum. We're not really reaching your colon with the celiac artery or celiac trunk, whereas your superior mesenteric artery and your inferior mesenteric artery are, the superior is this one up here, 8, and then your inferior would be down here. Those are those two big arteries that are supplying that anastomosis. So if anyone has questions about that, you can throw it in the chat and we'll try to answer. And then we can move on to the second question, which is kind of throwing back to some of the accessory organs we talked about. So a mass on the blank of the pancreas would cause compression of the duodenum. And then we just have all 4 sections of the pancreas.
All right, and for the sake of the time, I'll go ahead and close the poll in 5, 4, 3, 2, 1.
Sorry, I kind of clicked the answer there at the end, so some of you guys may have seen, but it's going to be your head of the pancreas. So just for the sake of not having to back up through my entire presentation, I'm going to try to see if I can go really quick, just to show you that picture of the pancreas again. But if we have a mass so we're talking about a mass on the pancreas that's causing compression of the duodenum. I went too far. Of the duodenum. So if we look at our pancreas right here, the part that's right next to our duodenum is going to be the head of the pancreas. So a mass on the head of the pancreas can actually sometimes present, patients will have, stomach pain. Sometimes they can have jaundice, problems with bile concentration and stuff like that, because you're pressing right on that duodenum and also that area, that ampulla of Vater. So yeah, that's how that can present. Any questions? I think that was the last slide, besides the references. Yep.
Great job, Calista, and thank you for that. While people are thinking of questions to either put out there or put in the chat, one thing I wanted to put out there about thinking through how should you approach the arteries and how should you study that is don't think too hard about it. Some of the names can tell you where they're going, like right colic, for example, is telling you that it's on the right side of the colon. So if you know that the ascending is on the right side, that's telling you, what side that's going to be on. So the names can be helpful. The other thing, as far as what they are supplying, there's a saying that we have in anatomy is if it's nearby, it will supply. So don't worry too much about sitting and memorizing everything that's being supplied. I think it's better to just understand approximately where the arteries are, and then you can sort of work them out for there. So I wouldn't sit and memorize, that'd be way too much for you guys to sit down and do. I see Sammy has a question for Calista.
Hey, yes. Two things to what you just said. I have a question. So, well, obviously I have a question, but if you do understand it, can you still memorize, or would that not be the smart idea to do so?
You can still memorize. I don't think it's the best tactic moving forward. I know it gets you through high school and probably through some of your undergraduate classes, but I'll say working through it, if you can understand it, I think that would show that you've done that. So you've made it through to that kind of proficiency and understanding. So sometimes you got to memorize to get to understanding. So but if you're to the understanding point, then you're in a good place.
Okay. And my other question, since I'm so big on cardio, um, I just recently learned about the IMA and SMA, and what problems would have to occur if, because I know I think it's AAA surgery that it happens with, but what problems would it just be like a bleed in there, or would it just be a bleed in them that would cause the need for surgery, or just what um, dealing with
Yeah,
the
I'm not gonna go.
Calista,
oh, so,
what have you had in what have you learned about in medical school so far that you can kind of key in
Um,
on?
So when you're talking like about like a abdominal aortic aneurysm, that's more what we learned is like a tear in the aorta. So that isn't necessarily a problem with your SMA or your IMA. That's kind of the entire aorta tearing. But some problems that could come from having like a blockage of blood flow with your like SMA or IMA is you could get a part of like we talked about something in class called like a um oh my gosh, what's it called when the blood isn't flowing there? Infarction, like you can kind of get an infarction of parts of your bowel if the blood supply is cut off there, and things that can cause that are, it varies. There can be kind of twisting of the gut in the wrong way, like a volvulus or an intussusception, or you can have just like a blockage, something like a mass growing. Does that
thank
kind
you.
of help?
Yes, ma'am. Thank you.
Okay, I think that will finish our anatomy discussion. We will move on to histology, and we're looking especially at the histology of accessory organs of digestion as well as the GI tract. And presenting for us this evening is Hannah Bash. Hannah is from West Hartford, Connecticut, and graduated from Washington University in St. Louis in 2022 with a BA in cognitive neuroscience and minors in Spanish and biology. She is currently a second-year medical student at the Cindy Kimmel Medical College at Thomas Jefferson University with an interest in surgery. Hannah enjoys running, cooking, spending time with friends, and her dogs. So with that, Hannah is all yours.
Sorry, can you hear me? Okay, perfect. Hi, everyone. Once again, I'm Hannah. Really excited to be here and be able to help with the histology session for the GI system. So we'll just go ahead and get started.
Hey, Hannah, I'm sorry to interrupt really quick. Do you mind changing your presentation mode? I think we're seeing the presentation view.
Is that better?
Perfect. Thank you.
Okay, perfect. Okay, awesome. So our lecture objectives for today, we're just going to describe and identify the 4 histological layers of the gut tube, and then we'll describe the components of the enteric nervous system and explain what the function is of that nervous system and be able to see them on our histology slides. So just to start off, a really broad overview of the gut tube and what it is. So the gut tube is part of our developing embryo, which I'm sure Kyle will go much more into in his embryology presentation on the GI system. But it's developing in our embryo and it forms part of our digestive system. And in our gut tube, we have 4 layers: a mucosa, a submucosa, the muscularis, and then a serosa or an adventitia layer, depending on where we are in our digestive system. And we can see these layers in many organs, such as the esophagus, the stomach, the small intestine, and many other organs in our digestive system. In my presentation specifically, I'll be showing you guys histology slides of the duodenum just to keep it continuous throughout the presentation so that you can see the same slide showing all the different layers of the gut tube. But just keep in mind as I go through the presentation that these layers do exist in other organs, not just the duodenum of the small intestine. So just keep that in mind. It's in all the organs of the digestive system. So to start off, we have the mucosa, which is the innermost layer of our gut tube. And if you look at the diagram on the right, our mucosa is what I highlighted in blue. So that inner layer that surrounds the lumen, which is the opening of the gut tube, and that's where food particles and stuff like that would pass through. And the mucosa is made of 3 layers. So we have an epithelium, which is the innermost layer that would be surrounding the lumen, and then a lamina propria, which is made up of loose connective tissue, and that's where we find blood vessels as well. And then we have muscularis mucosa, which is a smooth muscle layer, and that would be the deepest layer of our mucosa. So to look at it a little bit on histology, we'll do zoomed out pictures and then some more zoomed in pictures so you guys can see, but we'll be looking at the duodenum here. And where my yellow line is highlighting is the mucosa layer itself. And then if you look on the right a little bit further in, if we can get a more zoomed in image of what the mucosa would look like. So the lumen would be all of the white surrounding this. And then our mucosa is that big yellow line. And we'll go to another slide so that we can see the differentiation of the layers. Okay, perfect. So starting off, we have the epithelium, and that would be all around. And this is, for the example of the duodenum, a simple columnar epithelium. So really simple columnar is just those tall, skinny cells. And it's simple, meaning just one layer, versus stratified. You could have multiple layers of these cells, but simple, we just have one layer. Usually they should be aligned very nicely, as you can see in the more zoomed in image. They should line up pretty close to each other in parallel. And then we have our lamina propria, which is on the one layer further in, and this is made up of our loose connective tissue. And this, we can have blood vessels here as well, so we can see some capillaries, and that will provide blood supply to the epithelial layer. And then we have our muscularis mucosa further down, and this would be our smooth muscle layer. So a little bit smaller. As you can see, the lamina propria is probably the biggest layer that you see. And the epithelium can change too, depending on where you are in the GI system. This is just an example for the duodenum, but you can have different types of epithelium. It won't always be simple columnar, so just keep that in mind as well.
So Hannah, it's Dr. Peterson.
Oh,
Before you move on, because I think this is a really nice slide.
Yeah.
So Calista mentioned how the gut tube has increased surface area for mostly absorption. And she talked about those folds, and you can see a really nice picture here. Could you just maybe use your cursor and show everybody what we mean when we talk about the villi, those finger-like projections? That would be awesome.
Of course, yeah, thank you. Yeah, so here we are. As Calista said, it's really important to have absorption in our GI system. So these nice finger-like projections, if I just move my cursor around, you can see that is a villi, and then we have the nice fold that it creates, and then it continues on the other side. So around, if we go back, you'll be able to see. You can see all around multiple villi creating these folds all around. Okay. And then the submucosa is our layer just beneath the mucosa. And this is composed of dense irregular connective tissue, and then also blood vessels, lymph vessels, and nerves. And then a big part of the submucosa is glands. Depending on where we are in the digestive system, the glands will have different names. But usually if you see glands, that's a really big indication that you're in the submucosa. So that's the best giveaway on histology to know what layer you're in. So if we look at that a little bit further, same picture, we're just moving further down in our layers. So underneath our mucosa layer will be the submucosa, extending by that yellow line. And if we zoom in more, you can see the glands that I was talking about. So these glands should clue you into the fact that you're in the submucosa. So always keep that in mind if you see glands. And then on the bottom here, we can see our dense connective tissue. If you can compare it to the loose connective tissue we saw in the lamina propria, this dense connective tissue is usually more pink because there's more collagen there. It looks a lot more put together than our loose connective tissue, which is more like whirly, kind of pillowy, versus this is a lot more organized in its nature. And then if we go just a little bit further, specifically to the duodenum, we would call these Brunner's glands, but that name is specific just to the duodenum. So if you see glands somewhere else, it might not be called Brunner's glands, so just keep that in mind. But glands are always the giveaway of being in the submucosal layer. And then as we move further down, that dense irregular connective tissue layer, and just an example of some dense irregular connective tissue on the side over here. And then just a quick practice question to make sure that we're keeping in all of our layers in check. So this asks which layer of the gut tube is the innermost layer and then is further subdivided into 3 layers.
All right, and you guys are doing so great with participation on this one. I'll close the poll in 5, 4, 3, 2, 1.
Awesome. Okay, looks like most of the participants selected answer A, which is the correct choice, the mucosa. So our mucosal layer, if we recall, is the innermost layer surrounding the lumen of the gut tube, which is that opening of the gut tube. And then it is further subdivided into 3 layers, which are the epithelium, the lamina propria, and then the muscularis mucosa layer, which is our muscle layer. So great job, everyone. And then as we go further, we'll go into some of those other layers as well. And then there's one more question. What layer of the gut tube would this be where my yellow line is? And then we'll do the bonus question after. You guys could just type in the chat if you have an answer. Okay, awesome. I see some people putting in, yep, the submucosa layer. So as I said before, usually the glands are the dead giveaway that you're in the submucosa. So always keep that in mind. It's the easiest way to identify this layer on histology. And then for a bonus question, if anyone knows what these glands are called. Oh, I see someone put it in already. Yeah, the Brunner's glands. Yeah, exactly. So we're in the duodenum here. So these would be Brunner's glands. Great job. You guys are really smart. Perfect. Okay, and then we'll move on to our next layer of the gut tube, which is the muscularis layer. So this is composed of 2 muscle layers. We have an inner circular layer and then an outer longitudinal layer. Really important here not to get confused between the muscularis mucosa, which we saw in the mucosal layer as one of those subdivisions, versus this is just a muscularis layer. Some people call it the muscularis externa. So just remembering not to get those layers confused. Our muscularis mucosa here is within our mucosal layer versus this itself is a muscularis externa layer, and it has 2 submuscular layers, our circular and our longitudinal. So inside our duodenum, we can see as well here, just moving further down in our picture, nice division between our 2 muscular layers here. So over here, we have our circular layer and then our longitudinal layer on the outside. So as we go further to a zoomed in picture, we can just look at these again. This is our inner circular layer. So in cross section, it would look like this picture on the left. So our smooth muscle cells kind of bunched up versus our longitudinal section would look much more parallel, which is how you see it in the picture here. It looks like a bunch of parallel lines with the cells is usually the easiest way to identify that that's our outer longitudinal layer. And then our last layer of the gut tube is serosa and adventitia. This one gets a little tricky because depending on where you are in our GI system, some organs will have serosa. Depending on the part of the organ, it would be serosa versus adventitia, so that can get a little tricky. But to keep it as simplified as possible, it's our outermost layer or most superficial layer of the gut tube, so all the way out here. And serosa is just a single layer of squamous epithelium. found on our intraperitoneal organs. And then adventitia is usually a dense layer of collagen found on retroperitoneal organs, such as the esophagus. So retroperitoneal being organs that are more enclosed and behind other structures. And then a nice little mnemonic, serosa slides versus adventitia adheres. Usually collagen, you can think that that's adhering to something. So adventitia adheres, you'll see a collagen layer versus serosa is just our single. Also, you could use S and S, serosa, single layer of squamous epithelium. To remember that if you see just a single layer of squamous epithelium, that would be serosa versus adventitia is when you see all of the collagen. So we'll look at that in the next slide. So back to our trusty duodenum for the last layer that we see here is the adventitia. Lots of pinks. You would notice that this is all of the collagen, making this adventitia instead of serosa. If it were serosa, we would just see one line at the bottom of a nice squamous epithelium. But here we see lots of collagen, so that would indicate that it's adventitia. And then just a quick practice question to make sure we're identifying the layers correctly, to know which one represents serosa or adventitia here.
Alrighty, I'll close the poll in 5, 4, 3, 2, 1.
Awesome. So this one definitely tripped people up a little bit more. D is actually where our adventitia is over here. This is our outermost layer. C is actually our muscularis externa, so where we have the two layers, an inner circular layer and then an outer longitudinal layer. It can be confusing because they somewhat look similar, but in our adventitia with the collagen, you won't notice cells as much. So here in our muscularis layer, we see all of these dots, which are the nuclei and the cells of the muscles. So that can kind of clue you into the fact that you're in the muscularis externa layer versus out here in our adventitia, this is just collagen.
Hannah, it's Dr. Peterson again. Maybe with your cursor in the adventitia, you could just show them those rounded in cross-section blood vessels. We don't have to know whether they're arteries or veins, but that's another easy way to get a sense that you are in the adventitia when you see those larger blood vessels.
Awesome. Yeah. So if you guys can see my cursor right here, we have some blood vessels, definitely too far out to tell if they're veins, arteries, capillaries, but blood vessels can also be a nice clue to let you know that this is the adventitia. So all of these right here, different blood vessels that we have. Awesome. And then to go into the enteric nervous system, just to round out the histology of the GI system. So our enteric nervous system is the nervous system of our GI system. So it has its own nervous system that will regulate the passage and absorption of food. And it's further broken down into two plexuses, as we would call the submucosal plexus, which can also be called the Meissner's plexus, and then the myenteric plexus. So maybe based on the name, you can guess where our first one is going to be. But as we go, our Meissner's and submucosal plexus, however you want to call it, will be found in our submucosal layer. So once again, that's our second layer of the gut tube. And this will control secretions. So that's a really important function of this layer. So controlling secretions and blood flow. And then our myenteric, or also called the Auerbach's plexus, so you know when you see it written either way, this is found in our muscularis layer, so that muscularis externa layer where we have usually an inner circular layer and then an outer longitudinal layer. This can be found in between those muscular layers. So as you see right here, this nice nerve plexus coming in between our inner circular layer and then the outer longitudinal layer. very nice nerve plexus there. And this plexus is really important in controlling our smooth muscle contractions that will allow for motility of food in the gut tube. So every time we have a smooth muscle contraction in the gut tube, this will push the food bolus further along in the process and then allow that food to move down throughout the GI system. And then just closer up in this picture, you can see our inner circular layer at the top, the outer longitudinal layer in the bottom, and then the nerve plexus running right between them. So that would be our myenteric plexus or Auerbach's plexus, however you like to call it. And then just a quick practice question about our plexuses.
We'll go ahead and close this poll in 5, 4, 3, 2, 1.
Okay, awesome. So the right answer is A, which you see about almost half of you got. So our Meissner's plexus, which can also be called the submucosal plexus to give it away, is found in our submucosa layer, and it will regulate secretions and blood flow. For the other answer choices, I know I put some confusing ones in there. Found within the muscularis externa would be our myenteric plexus or the Auerbach's plexus. And then that will regulate motility. So that would be answer B. And then just switching up the layer on you, C would be partially correct because our submucosal plexus does regulate secretions and blood flow, but it's not found in the mucosa layer. So it would be in the submucosal layer. And then D, also partially correct. This is found in the submucosal layer, but it doesn't regulate motility. That is the function of the myenteric plexus. So just some quick answer choices there for you guys. And then there's one more histology practice question. So what plexus do we see in the image here? So there's a more zoomed out picture on the left and then closer up on our right side. And you can just type in the chat. Oh, sorry. Trying to get back to the chat. If anyone has a guess, and we labeled the layers to give them a little bit more of a hint there on where we are looking at. Okay, I see a few answers in the chat, so I'll just jump in. I see that people are writing submucosal plexus, which is a good guess, but unfortunately, we're actually looking at the myenteric plexus. So the adventitia here on the outside is labeled to kind of help you guys. The layer after the adventitia closest into our gut tube is our muscularis externa layer. So we can see right here, we have an outer longitudinal muscular layer, and then closer up here, our inner circular muscular layer. So in between our muscularis externa layers, we would see a myenteric plexus, which is what we're looking at. Our submucosal plexus would be closer up in this region where we have the mucosa on the outside is right here. So that's surrounding the lumen closest. And then underneath our mucosa would be the submucosa. And if we saw a plexus in here, it would be the submucosal or Meissner's plexus. They're actually usually very hard to find in histology images. You mostly only see myenteric or Auerbach plexuses, if that helps you in any way. But usually it is really hard to find a submucosal plexus on imaging. So if you see one, it's probably a myenteric, but make sure you're looking at the outside to see if you see serosa or adventitia, that usually clues you into the fact that you're in the muscularis externa layer where you see that plexus, and then that would be the myenteric plexus. And then just references and any questions at all. I know histology can be really hard. You guys are really amazing for doing this because I didn't even touch histology in high school, had no idea what that was. I don't even think we talked about it in college really until I got to med school. So this is amazing that you guys are getting such a head start on it. You'll be very prepared later on.
I was just going to say thank you, Hannah, for making me fall in love with histology again. I think that was wonderful.
Thank you.
Okay, I don't see any further questions, so we'll move on to our final presenter this evening, which is Kyle Yang. Kyle is going to be covering embryology of the gastrointestinal system. So a little bit about Kyle. He is currently a second-year medical student at Drexel University College of Medicine. He attended UCLA for undergraduate and majored in biochemistry. In his free time, he enjoys watching movies, playing games, and sleeping. He also enjoys traveling whenever he gets a longer break from school. His specialty of interest is emergency medicine, but he is open to exploring other specialties as well. So feel free to reach out if you have any questions. With that, Kyle, the floor is yours.
All right. Good evening, everybody. Thank you for being with me here today. Sharing my screen and slideshow. Let's see, annotate and give this a, see if I can do this here. All right. Oops. Sorry. Give me 1 second. Screen's being weird. Okay. I'm just gonna go here for now. Okay. Move this off to the side here. Okay, so I will be covering GI embryology today. And for our lecture objectives, we're going to be listing the organs that are derived from the foregut, the midgut, and the hindgut portions of the gut tube, plus a couple of extra things here and there along the way. Okay, so just a quick review from last week embryology. During week 2, after fertilization, we have our bilaminar disc, which starts out with an epiblast and a hypoblast with a primitive streak. And then by week 3, we have a trilaminar disc formed with the ectoderm, the mesoderm, and the endoderm. And this will be between weeks 3 and 4. And then folding really happens in week 4, and this is where we're really going to pick up our story with GI embryology. And so between week 3 and week 4, once the folding starts, you have a longitudinal view here. And then let's just imagine you sort of take a cross-section view of this longitudinal view right down the middle. You would see something like this. And so at week 4, you have your gut tube, which is made primarily of endoderm, and then you have the splanchnic layer of your gut tube, which is primarily made of lateral plate mesoderm. And then in general, your endoderm of your gut tube is responsible for the glands and the epithelium that we just covered in histology, and then your splanchnic mesoderm, your lateral plate mesoderm, is responsible for the smooth muscle and the connective tissue layers of your gut tube. And with that quick review, we just want to do one quick poll question, our first one of the day. For embryology, what germ layers does the gut tube derive from? So we have ectoderm, endoderm, mesoderm, A and B and C. Oops, sorry. Well, I kind of gave away the answer, so thank you, Mary. I'm just gonna move on here for the time being. So what germ layers does the gut tube derive from? Those who picked B and C were correct. So looking back at the image I showed on the first slide, we sort of have the gut tube, and which is sort of made up of the endoderm and the lateral plate mesoderm combined. They will give you the 4 layers of your intestines as mentioned in histology. Okay, good job, everyone. Okay, moving on here. So before we start thinking about GI embryo, just sort of giving you a quick, sort of really simplified view of it. You're just thinking about a tube that extends from your mouth to your rectum, and that tube is mainly divided up into 3 parts. You have your foregut, your midgut, and your hindgut. And then there are 2 really sort of major landmarks we use to distinguish where does the foregut end versus where the midgut begins, or where the midgut ends and where the hindgut begins. And that would be the major duodenal papilla and the about 2/3 of the way through the transverse colon. And we'll get to those in the further slides. Okay, so focusing up a little bit more on the primitive gut tube here, we want to realize that regionalization really begins at week 5. Around this point, your gut tube is suspended from the dorsal body wall, and so dorsal is sort of the backside. So the backside of the body wall, your gut tube is suspended from the backside with a suspension membrane, which will eventually develop into the major blood vessels and nerves that will supply sort of your GI system. And then cell proliferation begins between weeks 5 through 8, which leads to a gut tube being too long. And it's kind of shown here, but we have a better depiction of it in the future slides. But it ends up kind of being a little bit too long to fit inside the embryo's body, and it sort of grows into the umbilical cord a little bit before sort of retracting back. Okay. And so more on this gut tube rotation. So we have a picture here for you. So in picture A, we really see that the gut tube begins elongating, cell proliferation happens, and it sort of elongates into the umbilical cord. Just imagine that right around here is the cutoff where the umbilical cord is, and the tube is actively growing into the umbilical cord. In picture B1 here, this really cool thing happens where it actually rotates. The cord actually rotates, right? Because when we sort of look at your GI system as a whole, it's sort of all curvy and it has all these curves in it, and it really gets that right here in embryology when it starts rotating. And so B1, it herniates into the umbilical cord and it rotates 90 degrees. In B2, you're really thinking the inferior aspect of the tube really grows and becomes coiled, and it's depicted here where it starts coiling. And then in C, you see, or yeah, in C, it sort of retracts from the umbilical cord and it sort of rotates another 180 degrees, eventually creating something like you see on the right here, which is more indicative of what you would see in a sort of infant, or as you grow up, what your digestive system would look like. Okay. And then, so the coiled part of the gut tube is really centrally located, and the straighter and larger tube surrounds the central intestines as the colon by week 12 post-fertilization. And there is a short GIF here, however you pronounce it, that I would like to show you all. And so your fetus, you're taking a cross section here. You see your gut tube. It really herniates into the umbilical cord and it starts rotating, and it will rotate, rotate, rotate. And then once it has rotated, it starts retracting back into the embryo. Okay, to form the GI system that you normally think of when you think of a fully developed GI system. Okay. Okay, awesome. Slideshow here. Okay, moving on to the next slide, we have our second poll question here. How many degrees does the elongated gut tube rotate in total? So we have 90, 270, 360, or 180. Okay, we're still getting some answers in. I'll move on at 45 seconds here. Okay, 45 seconds, and it looks like the majority of you guys picked B, which is correct. It's 270 degrees. So how do we get that number? So like as I was mentioning in the earlier picture here, you have a 90-degree rotation in picture B1, so it's 90 degrees when it is in the umbilical cord, and then in picture C, once it retracts, it rotates another further additional 180 degrees. And so we get 180 plus 90, you get a total rotation of 270 degrees in complete. Okay. Moving on to focus a little bit more on the foregut here. I'm sure it was mentioned before already, but your main structures that come from your foregut are your esophagus, sorry, depicted by the structure labeled 1, your stomach labeled 3, your liver with 4, pancreas with 7, your proximal duodenum of your small intestine with 6. And this is sort of everything before the major duodenal papilla, which is that landmark that I mentioned earlier that separates the foregut from the midgut. Okay. Moving on a little bit to the development of the foregut structures. I want to focus on sort of the bile duct, which was mentioned earlier in anatomy. It sort of connects to your liver and your pancreas and your gallbladder. And so your bile duct sort of grows from the primitive gut tube right here, right at the location of the major duodenal papilla. Your gallbladder, your liver, and the ventral part of your pancreas will develop from the bile duct. Meanwhile, the dorsal or the sort of more backside part of your pancreas will develop on the other side with the dorsal pancreatic bud. And so, and it grows directly off of the gut tube. And then as the gut tube rotates, like we were discussing earlier, it sort of rotates the ventral pancreatic bud, it sort of brings it in line with the dorsal pancreatic bud, and then they fuse to become your complete pancreas. And I think that's really cool here that it can rotate like that. Okay. Moving on to the structures of your midgut. So as I was mentioning earlier, again, your midgut sort of spans everything from the major duodenal papilla to about 2/3 of the transverse colon. And so when we look at the picture of your colon here, it's really divided into 4 sections. You have your ascending colon, and then it curves at the hepatic flexure because your liver is right on this side, to your transverse colon, and then it curves again. This is called your splenic flexure because your spleen is right on this side, and finally becomes your descending colon, your sigmoid colon, and your rectum. And so right around where this dotted black line is sort of the cutoff between your midgut and your hindgut structures. And so your midgut is really only responsible for, as you can see, about the first 2/3 of your transverse colon. In addition to that, it covers your distal duodenum, because if you remember, the foregut only really covers the proximal part of your duodenum. Your midgut does the distal duodenum, your rest of your small intestine, which is made of the jejunum and the ileum, and then, of course, the colon, like we were discussing. It covers the cecum, the appendix, the entire ascending colon, and 2/3 of the transverse colon. Okay. And that will bring us on to poll question number 3. So what structures would be missing if the embryonic bile duct failed to develop? Would it be your liver, your spleen, your gallbladder, A and B, or A and C? Okay, get your answers in just like before. I'll probably move on at 45 seconds.
Be in 5, 4, 3, 2, 1.
Okay, awesome. So we have a good spread of answers, but the majority of you chose E, which is correct. And I see there was a couple choices with A and C, which are correct as well. And so moving from the picture shown here, you see just an earlier picture from an earlier slide. We see our primitive embryonic bile duct, and we said that your liver, your gallbladder, and the ventral part of your pancreas, a pancreatic bud, will develop from your bile duct. And so liver and gallbladder is the correct answer. Your spleen actually develops from a different part of your embryo. Okay. Finally, moving on to the hindgut, as I mentioned already, it's everything after the 2/3 transverse colon mark. So everything past this dotted line here. So it's the last portion, your last 1/3 of your transverse colon, your descending colon, your sigmoid colon, and your rectum. Okay. And then so sort of we have our summary here. We have our foregut, our midgut, and our hindgut. And I want to sort of use this slide to bring a little bit more attention to the blood supply. Blood supply to the GI system, I think, is a little bit less complicated than blood supply to many other parts of your body. Essentially, your foregut is entirely supplied by branches of your celiac artery. Your midgut is entirely supplied by branches of your superior mesenteric artery, and then your structures of your hindgut are completely supplied by blood supply from your inferior mesenteric artery. Okay. And sort of this is sort of like a very typical question that I remember being asked when I was learning about the GI system. And it was essentially just asking what major blood vessel supplies blood to the transverse colon. And so we have our answers here. We have our inferior mesenteric artery, superior mesenteric artery, celiac artery, and it depends. And so okay, once again, we'll probably stop at around 45. So in about 5 seconds here. 2, 1. Okay. And so a majority of you chose A, which isn't 100% wrong. It is correct. But as I was mentioning earlier, so your midgut does do a good part of your colon, including the ascending colon and 2/3 of your transverse colon. But that little 1/3 in the back there is technically a structure of your hindgut, right? And as I mentioned, all the structures of your midgut come from your superior mesenteric artery, and then all structures of your hindgut get their blood supply from the inferior mesenteric artery. And so we have to realize, and this is very important here, that your transverse colon is actually derived from both sections, your midgut and your hindgut, which have 2 different blood supplies. Okay, and so which ones? It would be your inferior mesenteric artery and your superior mesenteric artery, as I discussed earlier. Okay, and these are my references. And are there any questions? It's just a very short intro into GI embryology here, and most of my work was done for me by the 2 awesome previous presenters. They really laid the foundations of everything else.
Question.
Yeah.
I know you're like, of course he has a question. But what are the most gastrointestinal bleedings, and how do you see them in like a presentation?
That's actually a good question. I actually haven't seen any GI bleeds yet. I know we've done our anatomy lab, but there's no active bleeding going on there, so I might actually have to pass this question on to maybe Dr. Peterson or one of the faculty.
I haven't seen them either yet. That's why I was like, man,
Yeah.
if somebody else has seen it.
So Sammy, I just didn't hear your question. Can you just repeat it?
Yes, ma'am. Yes, ma'am. What are the most gastrointestinal bleedings, and um, how do they differ in presentation?
I can kind of jump in with just a really vague answer to your question because it really depends. One thing that's kind of interesting would be where the blood is coming from, right? So if someone is vomiting up blood or coughing up blood, right, it's likely to be from like the upper GI tract, from the stomach or something like that. But then if you had blood in your stool, right, it'd be more likely to be below that pyloric sphincter, down more into the intestines, that the bleed would be coming from. But gosh, there's so many different reasons why your GI tracts can bleed. But that would be kind of one thing that if someone showed up, in the ER and they saw blood in the toilet, right, that might be the instigating reason why someone would then kind of undergo an investigation of where that bleed is coming from and why it's happening.
So that would cause hematemesis or hematypesis?
Yes.
Okay.
Okay, good question, Sammy. Are there any other questions about sort of the embryology of the GI tract?
I think we had one good one in the chat. That was from Angel. If the liver is so huge during development, does it get less ambitious after birth? Why does it shrink in proportion to our body as we grow? Dr. Frank had an answer in there. Kyle, if you'd like to read it off, or if you'd like to put your 2 cents in there, but just for the people that are attending this afterwards that can't read our chat.
Actually, I can't read the chat either. I actually can't see the chat. But I actually can't see the question either. Let me see if I can. Dr. Frank, do you mind reading it off? I really can't see it, or I don't know how to
Okay,
get it to show up on
no
screen.
problem.
Oops.
If the liver is so huge during development, does it get, quote, less ambitious after birth? Why does it shrink in proportion to the body as we grow?
I don't think the liver shrinks, right? It grows, and it's a massive part of your stomach cavity.
I think they're maybe talking in proportion to the size of the infant. The liver is quite large at birth comparatively, and as the body grows, the liver doesn't keep up at the same pace as the body and becomes less in proportion as the body grows. It can also shrink with age, and there are other pathologies. One in particular would be cirrhosis of the liver, that can also shrink the liver. Remember that the liver is one of the organs that can regrow.
Right, and I'll
So,
just related to embryology, Dr. Frank, and I know, you have presented this to our Drexel students, and more than a few times. But the liver also, Angel, and this is something that's really, you're not going to be tested on, but it's just a cool, fun fact that after the embryo gets beyond about when it has a fully developed liver by week 8, it will take over the function of what early on in embryonic development were those blood islands that we saw on the surface of the yolk sac. Remember, those were making red blood cells and white blood cells. Well, after about week 8, when the, again, liver is at least in some rudimentary way completely formed, it will take over that function for the developing fetus up until about month 7. So it has to be relatively large because as the embryo grows, it needs more and more of those blood cells. So that's a very cool fun fact.
Yeah. Thank you, Dr. Peterson. And I noticed, Sammy, I think you had your hand raised again.
Hey, yes. So this is about the studying.
Mhm.
I've been studying everything for our upcoming local test, and I was wondering, is it going to be everything that we've learned from 1 through everything we finished, or so we need to be looking at all of that, everything that we have learned from, should we be studying all that material over and over?
Yeah,
'Cause
so
that's what
it will be all the content for specifically the local learning objectives. So if you look at that sheet that Dr. Haggerty pulled up for the learning objectives, there's some of the regional objectives that I think we've kind of tickled on a little bit and kind of go a little bit more in depth at times. But the most concise answer I can give you is that if you can answer the learning objectives in there, then you're in good shape.
Okay. Also,
Dr. Peterson has something to add as someone who also.
okay.
Okay.
And so last year we did the patient. You remember the patient thing with So are we gonna do that again so I can be a little more? Okay. Okay.
Yeah.
Do that.
Yeah, we're hoping to get more details for the regional competition to you guys soon and we'll make those widely available to everyone on our website when we
Kind
do.
of lock in again. So
Yeah. Yeah,
Thank
and just to say, Sammy, we appreciate all your questions. They're very inquisitive, and so don't be afraid to ask questions. We're glad that you're here.
Thank you. They helped me during rounds, so y'all helped me a lot. Y'all helped me a lot, so thank you.
Good, good. And then I think there's another one in the chat, Kyle. Another really good question with Angel that I think is just really thinking outside the box here is if bile starts forming before birth, but digestion isn't active yet, is the gallbladder like a storage closet that babies don't even need?
Well, they need it at some point. So I mean, you wouldn't be wrong, but we grow all of the stuff that we have basically in birth, right? So we do need it at some point, but yeah, I think you'd be right.
And it's part of the reason why the baby's very first poop, Angel, is kind of really thick and kind of has the consistency of tar. And some of that dark color comes from the bile that has been sitting in the gallbladder for so long. So that's when people talk about the meconium, that's the first baby's poop, and that's why it's so dark and tar-like in color.
Then there's one question that I think we can help answer from the anatomy standpoint is if there was a tutoring session for the standardized patient diseases. One thing Dr. Haggerty mentioned at the start of this was that we'll do a review session for the local objectives. You guys will get familiar that that's going to be us just basically the same way that we've been doing interactive questions during the lecture. We're going to have some that are kind of targeted in that way, and it's going to be a very interactive session the whole entire way through. When we get to the regional session where you actually would be responsible for those standardized patient cases, we will go through an example of those. And then as well as when we put that information on the website, we'll put another example on the website as well. So you'll get at minimum 2 examples to work through for those standardized patient cases. And then a really good question here from Eliam is, why do some people end up with 2 gallbladders? Is it like having a backup hard drive, or does it make digestion more confusing? I like these analogies. That's so great.
Yeah, so like occasionally during birth, something fails to divide. And another analogy I can think of is for your urinary system, right? Sometimes you can actually have 2 ureters because the structure that it derives from fails to divide. And so you can think of like a second gallbladder almost like just not like something major went wrong during development, and I wouldn't even really call it going wrong because you can function normally, right? You just sort of have a backup gallbladder. Whether or not it sort of impacts your functioning really varies on a case-by-case basis. Sometimes I think you could just have an extra gallbladder that's non-functioning and you'll be fine. And then maybe other times there can develop into a functioning gallbladder. So yeah, it really depends, but it is completely a possibility. But we really only need one. So good question. Okay. Are there any other questions, Mary?
I think that's it. I think we might be all done Or I got one more in here. We'll do this last one and then we'll let you guys go early. I know I'm still digesting all my Thanksgiving food and need some more rest, so maybe you guys are in the same boat. This question is, if the gallbladder is just a bile storage tank, why does it even need its own unique developmental pathway? Could the liver do fine without it?
So you can survive without your gallbladder, right? People have had their gallbladders removed. Um, as to why we have it, I'm not entirely sure, besides the fact that it just is a storage for bile. I think it's just another step to ensure that you have adequate secretion of bile.
And I'll just jump in and add, but more than just a storage tank, it's also concentrating the bile. And so that's really important as well. And people that don't have a gallbladder, they typically, and that's surgically, that's something that, they agree to let a physician do typically. So their bile is much more dilute and it's constantly being produced by the liver, and it just is draining or trickling down through that ampulla of Vater or Vader, pronounce it either way. So it's constantly in the duodenum rather than only when you have a really heavy lipid laden meal, which is when it gets signals from the stomach to actually secrete that bile into the duodenum. So, yeah.
Okay.
All right, so
Thank you guys.
yeah, thank you so much, Kyle. Final question here, is all the content taught in the tutoring session good for the anatomy local competition? Is there anything else in anatomy that is not taught here? Everything should be covered through the learning objectives that we have posted at the beginning. So as far as the local competition, I will say there's many times where we go beyond what you guys need to learn. So again, if you can concisely understand and sort of work your way around those learning objectives, then you'll be in good shape. So with that, I see we actually had one more question come in. I'm not sure, Dr. Beeson, you got time for one more? All right, we got the foregut rotates during development to put everything in its right place. What would happen if the rotation got stuck? Would we be walking around with livers on the wrong side or upside down?
I'll let Kyle give that a shot and then I'll jump in.
Well, I think it depends on the degree of stuck, right? Because if it's just a little bit and it's mostly done, I think you could still completely function. Everything inside would just be maybe a little bit misaligned, just a little bit. But I feel like if you had really early on, it just didn't rotate, I just don't believe it would be compatible with life. But I could be completely wrong.
And we've talked about this before. Good answer, Kyle. There are different scenarios congenitally where everything is rotated to the opposite side. So early on in embryology, there's some signaling factors that have to be made at the appropriate time in the appropriate concentrations so that all of the organs that are not paired so that they end up on unilaterally on the right side of the body that they're supposed to. And again, if things go wrong and that those signaling factors aren't there at the appropriate time, you can get this reversal of organs. And Dr. Frank just put some information about it in the chat. So yay, that's a very cool idea to end on. So it was great to see all of you. And for all of you out there watching the video, again, keep studying. And we're just really grateful, as Dr. Prospera said, for the time you're willing to spend with us. And again, the time that the medical students are willing to share with all of you. So thank you and have a good evening.
Can we remind them where they can get the videos for these sessions again?
Yeah.
A couple of people asked.
So if you just go to our website, Anato-Bee.org, then we post all of the videos within 24 hours of the session. You can go to our tutoring tab and scroll down and there'll be a link to the most recent uploaded video. And that's how you can get to our YouTube channel. Conversely, if you're on YouTube, if you just Google or search in the YouTube search bar Anato-Bee, then we should come up there as well. And you can see all the sessions. I will say, make sure that you're looking at this year's tutoring sessions because we've slightly modified some of the learning objectives from the previous year. And we want to make sure that you're studying all the right content for the 2025 competition. So just everything ditto to what Dr. Peterson said. Thank you for reminding us to say that, Dr. Frank. And we will see you guys again on December 15th. So everyone have a great evening and we'll see you next time.